Today I want to share an incredible breakthrough in medical science. In the UK, researchers have created a super baby with DNA from 3 different people, effectively giving the baby 3 parents instead of 2. This innovative technique ensures that the child will not suffer from mitochondrial genetic diseases, although it doesn't eliminate the risk of other genetic conditions. Many of you might not know that every cell in our body contains mitochondria, known as the powerhouse of the cell because they produce ATP, or adenosine triphosphate, the molecule that supplies energy. Mitochondria have their own unique DNA, separate from the nuclear DNA in the cell's nucleus. Research has shown that if this mitochondrial DNA has mutations, or if defective mitochondria from the mother are passed on, the baby becomes susceptible to mitochondrial diseases. This specifically involves the mother's DNA, since the sperm's mitochondria degrade at the beginning of fertilization. Thus, only the mother's mitochondria are inherited. Researchers conducted a study on women aged 26 to 42 to examine their eggs. They found that women aged 26 to 36 had mutations in just 1 egg, while women aged 37 to 42 had mutations in 6 eggs. This suggests that mitochondrial defects increase with age, raising the risk of babies being born with mitochondrial disorders for older mothers. Our body functions like a machine, and just like any machine, errors increase with time. During DNA replication, errors can occur in 1 base for every million bases formed. For mitochondrial DNA, this error rate is 1,000 times higher than nuclear DNA, mainly because mitochondrial DNA lacks the protective enzymes and proteins that nuclear DNA has. Consequently, mitochondrial DNA is more prone to damage, causing severe diseases like Leigh syndrome. Leigh syndrome affects Jessica, leading to brain shrinkage and slowed motor development. Sadly, she now needs to be tube-fed, and there is currently no treatment that can fully cure her. Doctors believe she might not live much longer because mitochondrial diseases can impact any vital organ such as the brain, heart, or kidneys, potentially leading to death. Among these diseases, vascular issues are the most frequent cause of death. For example, in the UK, over 150 children die each year due to mitochondrial mutations, and globally, 1 in every 5,000 people succumbs to these diseases. There is hope, however. Scientists have devised a way to repair these damages through a 3-parent baby technique. By studying mitochondrial processes, they found that the enzymes needed to make ATP are stored in mitochondrial DNA, not the nucleus. If these enzymes aren't produced timely, the body could stop functioning, as happened in Ben's case. Ben, an 11-year-old boy, had a genetic disorder called hypotonia, causing his muscles to soften and halt basic movements. At age 9, tests showed his kidneys were failing and there were multiple heart blockages. Despite immediate treatment, he unfortunately died of a heart attack. His condition was due to defective mitochondria producing only 2 ATP molecules, as the Krebs cycle and ETC, electron transport chain, occur in the mitochondria. To compensate for ATP loss, cells start lactic acid formation, which usually happens during intense exercise. This causes a buildup of lactic acid, making those with mitochondrial disorders feel like they are constantly exercising. If you've ever worked out and felt sore muscles, that's due to lactic acid buildup, which causes muscle fatigue. If this continues, especially in the heart, it can result in heart failure, as it did with Ben. Lactic acid releases hydrogen ions, disrupting the body's pH balance. To address the root of these disorders, scientists thought of replacing defective mitochondria with healthy ones from a donor using in vitro fertilization. When this was first tried in 1996, the results were troubling, as babies had developmental issues and genetic defects. However, trials on monkeys were successful. In 1980, scientists transferred cytoplasm between monkey eggs. After that, the method was approved for humans in 1996, but due to negative effects, the FDA banned it, leaving it theoretical. That changed in 2004 when the UK's Human Fertilisation and Embryology Authority developed 2 new techniques. By 2016, the first 3-parent baby immune to all diseases was born. The nuclear transfer technique involves 3 steps. Healthy mitochondria are extracted from a donor egg. Then, through IVF, eggs from parents and the donor are fertilized. 12 to 18 hours later, the nuclear material is extracted, and if healthy, it is transferred to the donor egg. This creates a healthy super baby. But this process has a drawback. The whole embryo is discarded, which, well, raises ethical concerns. Thus, a method called maternal spindle transfer was developed. It involves removing the nucleus before fertilization and transferring it to the donor egg during the metaphase stage, where chromosomes align, making it easier to spot defects. If the mother's mitochondria are defective, why not use the father's? The reason is mitochondrial population. A sperm cell has 50 to 75 mitochondria, while an egg has about 100 million. This makes the donor necessarily a female egg. This method has a 70 to 80% success rate, which means a high chance of a healthy super baby. Like everything, there are two sides. While IVF babies can be immune to genetic disorders, there are risks when transferring the fertilized egg to the mother's womb. Factors like maternal age, environment, and fetal health could cause complications. To prevent this, an artificial womb with all necessary nutrients has been designed for baby development. Artificial gestation is revolutionary, as explained earlier in this video. If you found this video helpful, please like it, subscribe to the Do You Know channel, and hit the bell icon to stay updated. See you next time. As always, stay curious and keep learning.